Welcome to our presentation by Jayan Park, Jeff Kim, Sophia Yu, and Ryan Lee. This is the structure of our modern society. Structure of the Earth, structure of the Louvre Pyramid, structure of our human ear. And now, what about the structure of DNA? Do you want to find out? 8.2 The structure of DNA. This is a human body. If you go in, you have cells. If you go further in, you have all of these DNA molecules within the nucleus of a cell. And if you go further, notice how each DNA molecule consists of nucleotides. And in this diagram, we have 4 nucleotides within the circle: a phosphate, a sugar, and a nitrogen-containing base make up one nucleotide. And notice how that the nitrogen-containing base is also referred to as nucleotide. And there are 4 types of nitrogen-containing bases: adenine, thymine, cytosine, and guanine. A, T, C, G. A, T, C, G. A nucleotide consists of one phosphate, one sugar, one nitrogen-containing base. And there are 4 types of nitrogen-containing bases: A, T, C, G. And notice how they are classified into 2 categories. Adenine and guanine are called purines because they have a double ring structure. Notice that they each have one hexagon and one pentagon. And on the other hand, thymine and cytosine are called pyrimidines because they only have one ring. Erwin Chargaff was active in the 1950s, and he proposed that all 4 bases do exist in all organisms, only that their proportion in the DNA differ from organism to organism. And he came up with one rule, which was that the amount of adenine equals the amount of thymine, and that the amount of cytosine equals the amount of guanine. So again, we have ATCG. ATCG. Now we'll examine some key figures. Watson and Crick were scientists, best friends, and they wonder what the DNA looked like. Rosalind Franklin, also a notable scientist at the time, kind of had a general idea on the structure of DNA, but of course, they were her rivals. So she did not tell them. And the method that she used was X-ray crystallography, in which she bombarded the DNA with X-rays, and the atoms in the DNA would diffract the X-rays so that you would have a pattern on the film. And the result of this, as can be shown in the photograph here, is an X-shaped thing surrounded by a circle. And this is exactly what happened when she beamed at the vertically suspended DNA. Wilkins was a co-worker of Franklin, but he kind of betrayed her by sharing information to Watson and Crick without Franklin's consent. Watson and Crick, using this information, using this X-ray crystallography, was able to come up with a model of DNA, double helix model. And they published this on the Nature magazine, and they said that the DNA is a twisted ladder because there were 2 strands of DNA winding around each other. And they're right. In fact, these strands were complementary, and this should remind you of geometry. And complementary meaning that they fit together and that they were the opposite of each other. And double ring nucleotides fit with single ring nucleotides. And we will now examine how the bases paired with each other. It's very simple. A goes with T and C goes with G. Again, AT, CG. And notice in the diagram that the bases are connected are bonded to each other by a hydrogen bond, which is generally weaker than covalent bond, and a covalent bond would connect a phosphate and a sugar. And between adenine and thymine, there are 2 hydrogen bonds, but between cytosine and guanine, there is one extra hydrogen bond bonding these 2 bases. And even the hydrogen bonds are weaker than covalent bonds, they are still supportive enough. And the backbone of the DNA is the alternating phosphate and a sugar anyway, so the bases won't matter. So if one strand of DNA reads GTACGA, the other strand of DNA would read CATGCT. That was the end of our presentation. Thank you.